and today I've decided to discuss this biological question for new curriculum. Uganda, it's not a question, it is an item for you guys who are sitting senior 6 this year, and this is my first question to attempt, and if it reaches 100 views, I'm going to post another question, and the trend will go like that. If I get 100 views, I'm posting another video. 100 views, I'm posting another video. That means now it stands your way to get these free videos for free, because some people are selling this as premium content, but I'm going to be explaining more than even your teacher in class would explain to you this biology. So don't forget to subscribe, don't forget to share this video to your friend, don't forget to like this video so that it moves to every student. And if I get my 100 views, I'm going to post another item too. So I leave that to you. Let me do my job. This question is saying like this: agricultural science, we are studying lettuce, a th3 plant, and if your minds, make sure your minds are now here. Now, if you think of a th3 plant, or if they talk of a th3 plant, just think of a plant which grows in those areas of low temperatures and high altitude. That must come in your mind. And a C4 plant, if someone talks about a C4 plant, what you must understand is that plant that grows in areas of moderate temperatures or high temperatures and low altitude. So now I'm here on a C3 plant. Now they are saying that in a controlled greenhouse, every word in biology has a meaning, and every word, they have a reason why they put it. A greenhouse, just not it regulates. A greenhouse, it is that house farmers use to regulate those temperatures, to regulate the conditions. They are regulating the conditions. Sorry for that. They are regulating the conditions inside the greenhouse. Sorry, why is this app opening? The greenhouse, the regulating conditions. That means temperatures are constant, and other conditions favoring photosynthesis are kept constant. Hope you know those conditions from senior 3, senior 2. From your O level, you know those conditions that favor photosynthesis. And they are saying in a controlled greenhouse, to improve the yield, they vary the light intensity. As you see, they are varying light intensity and carbon dioxide concentration. That means, let me first explain that line, that if you vary light intensity and also carbon dioxide concentration, because if you increase light intensity, that means the rate of photosynthesis is going to increase. And the moment you increase light intensity, you're providing more light energy to the plant, favoring high productivity and high yields, and the carbon dioxide concentration while keeping the temperature at 25. And if you increase carbon dioxide concentration, that means you're going to be increasing the rate of photosynthesis. This is how carbon dioxide it is used to reduce. It reduces those molecules in the light-independent reactions of photosynthesis. For example, carbon dioxide combines with Rubip to form S.6 unstable carbon compound. We don't normally talk its name, but that it is unstable to the extent that it immediately splits in two molecules or three carbon compounds called triose phosphate or PGA. And that PGA, through series of carbon rearrangements, it is also phosphorylated to regenerate the Rubip. I'm explaining the Calvin cycle right now because this question is about the Calvin cycle. Make sure your minds are on. If you didn't understand this topic in class, Now we are answering a question and we are preparing for your name. And I told you just I want 100 views for the next video. Make sure you do the needful. So, hope you have understood what carbon dioxide concentration does. Increase in carbon dioxide concentration provides more carbon dioxide molecules to combine with the BIP and increasing the photosynthesis rate. Now, after 10 days, they subjected half of those C3 plants, lettuce plants, to water stress. They are talking about water stress now. Not just a thing that they have just talked about it for formality. Now, water stress, what does water stress mean? Water stress increases the concentration of a chemical substance in the leaves called, and that chemical substance, it is called abscisic acid. Now, abscisic acid, for it stimulates stomatal closure to prevent excessive loss of water to the external environment. That means if the stomata is now closed due to the presence of abscisic acid, less carbon dioxide will be presented to facilitate the increasing rate of photosynthesis. That means the photosynthesis rate will decrease, right? Because we now have water. The plant is now undergoing a condition called water stress. There is increased concentration of abscisic acid. Stomatas are closing. The plant is no longer getting enough carbon dioxide. The concentration of carbon dioxide decreases. That means if the concentration of carbon dioxide decreases, we are going to have a decreased rate of photosynthesis, hence decreased productivity. Right? Make sure you note somewhere. Make sure you subscribe to this YouTube channel and make sure you share this with your friend. By reducing irrigation, because if you irrigate, you are providing more water. That means it is a condition favoring water stress. Now, they measured the rate of photosynthesis using a gas exchange analysis. The scientists noticed a sharp decline in the rate of photosynthesis under water stress. Hope I have explained why there is that sharp decline in the rate of photosynthesis under water stress. Let me explain it. Why there is a sharp decline? Stomata, because during water stress, stomata close and the plant no longer gets carbon dioxide from the atmosphere to combine with the BIP to form triose phosphate, which regenerates BIP through series of carbon rearrangement reactions. But now the biochemical analysis of the leaves showed that some of the triose phosphate produced during the Calvin cycle was used to synthesize the proteins and lipids rather than starch. Biochemical analysis showed that those leaves of some triose phosphate produced some of triose phosphate produced during the Calvin cycle was used to synthesize proteins and lipids rather than starch. I will explain how those proteins are produced and lipids are produced and how starch is produced in this Calvin cycle. But just to know that all those, they are polysaccharides from chemicals of life. Protein is a polysaccharide, lipids are polysaccharide, and starch, it is also a polysaccharide. This is now how we are going to, and this is now our task. As a biology student, they are saying, explain how changes in light intensity and carbon dioxide concentration affect the rate of photosynthesis, affect the photosynthetic rate of that lettuce seed. Now, I'm going to start with the rate of light intensity. Increase in light intensity. Increase in light intensity. It increases the what? The photosynthetic rate of that lettuce and vice versa. By providing more light energy, for the light-dependent reactions, full stop. That is how. Don't wandika bingi, wandika okukati. Increase in light intensity increases the rate of photosynthesis of lettuce, because, and vice versa, because increased light intensity provides more light energy during the light-dependent reactions. Remember, in the light-dependent reactions, we have the light-dependent reactions. They are of two kinds. We have those light-dependent reactions. There is those light-dependent reactions that involve only photosystem 1, and there are also those light-dependent reactions that involve both photosystem 1 and photosystem 2. In those that involve only photosystem 1, they are producing only ATP. And those that involve both photosystem 1 and photosystem 2, they are producing both ATP, NAD, and oxygen as a byproduct. I hope you know how oxygen comes about. There is photolysis of water. Okay, thank you guys. Now, don't forget to like, don't forget to share this video to your friend, don't forget to subscribe to my YouTube channel. I told you this channel is not about that, but subscribe, because I'm just testing you guys whether you can support me. I drop more content about this one. This channel it is a STEM channel: science, technology, engineering, and mathematics. So I want to also enter the curriculum of Uganda and explain, and I make some people pass this A-level new curriculum. And we also have the carbon dioxide concentration. How does carbon dioxide concentration affect photosynthetic rate of the lettuce? Just know that if you increase carbon dioxide concentration, the photosynthetic rate, I'm starting by describing, hope you are noting how I'm phrasing my points. Increase in the carbon dioxide concentration increases the photosynthetic rate of lettuce, and vice versa. Now, how and why? Because we are in explaining why does it do like that? Because carbon dioxide molecules combine with ribib to form S.6, to form triose phosphate. Full stop. Now they are telling us to suggest a way through which science could manipulate environmental factors to optimize lettuce yield in the greenhouse. Environmental factors, I hope you understand, you can differentiate between those environmental factors and the non-environmental factors. Environmental factors mean environment, things surrounding man. Okay, hope you understand now. Climate, we may have drought and famine. Now others, I didn't take geography. I don't know those environmental factors, but as a biology student, this is how we are going to go on with that question. Manipulation means changing or something of the kind, changing the environmental factors. How can you be changing the environmental factors to increase productivity in the greenhouse? The first step is that we can create irrigation schemes in the greenhouse. Now we are manipulating that factor of climate, because if we create irrigation schemes, it is a point. We are going to be providing more water to the plant in the absence of rain. Rain is a rainfall environmental factor. We are going to have an adjustable shading nets. We are going to have shading systems to regulate the amount of sunlight in case of excessive sunlight. We are going to have shading systems to regulate that amount of sunlight that enters the greenhouse, because increased sunlight may increase the temperatures in the greenhouse beyond that the optimum temperatures, which cause the denaturation of those photosynthetic enzymes. And the last point, I'm going to talk about the proper ventilation in a greenhouse. Hope you are seeing how my points are relating to the question: temperatures, environmental factors. Now we are going to regulate ventilation in the greenhouse. Ventilation, in this case, we are going to have the cooling system or cooling pads to prevent overheating in a greenhouse. Remember, you are going to have shading systems and the cooling pads. Those points, hope you get that now we are dealing with external factors. We are manipulating them, changing them. I said we are going to have cooling systems, we are going to have shading systems, we are also going to practice irrigation. And practicing irrigation provides more water directly to those lettuce roots. Shading systems, they regulate the amount of warmth in a greenhouse. Cooling systems, they also regulate the amount of warmth in the greenhouse. Now, we are going to describe how, the last part, describe how triose phosphate from Calvin cycle can be converted into proteins. I'm going to start with that one. Remember, proteins are polymers from organic chemistry. What is a polymer? You can pause the video and think about a polymer, and then we continue. And what is a monomer? We can also pause and think about a monomer, and we can also continue. But polymers, they are large molecules formed from combination of those small molecules. Hope now you can understand. This is how a protein is formed. We stopped. They are saying how triose phosphate from the Calvin cycle. Now, this triose phosphate, as you see it, this one, it is a 3-carbon compound. Now, it will polymerize and forms hexose sugars. Hexose means X from primary 7 hexagon. It is a 6-sided figure. Now, triose means from our primary 7 a triangle. A triose, now it means a 3-sided figure, triangle. But now we are dealing with biology. Triose phosphate, a 3-carbon compound, polymerizes. Now they are molecules. Triose phosphate molecules, they polymerize to form hexose sugars, which may include glucose and fructose. Now the glucose and fructose molecules, or hexose sugars, they are mobilized in the Krebs cycle and the glycolysis, forming organic acids, which may include oxaloacetic acid from the Krebs cycle, may include alpha-ketoglutaric acid also from the Krebs cycle. We may also have the pyruvic acid from glycolysis. And now we are going to have the nitrates absorbed by those roots, by the lettuce roots. They are going to be reduced to ammonia. Now we are going to keep that ammonia aside. Nitrates, they are absorbed by the lettuce plant roots, are going to be converted to ammonia. Now ammonia combines with those organic acids I have mentioned above, which may include alpha-ketoglutaric acid, oxaloacetic acid, and pyruvic acid, forming, they are going to combine. Just leave it as ammonia combines with organic acid. You're going to have now amino acid synthesis. Now ammonia, that with those carbon skeletons of those organic acid, we will be converted to amino acids, and those amino acids will now polymerize to form proteins. Let us go on how those lipids are formed. tusobola okuddamu buno oba protein gyamufuumingamu ne tukasamalizinga. Tugambye tulina ono triose phosphate ono. Triose phosphate, it is going to triose phosphate bagenda, it is going to be converted to it is going to polymerize to extra sugars, and those extra sugars will enter the Krebs cycle and glycolysis, forming organic acids, and those organic acids include pyruvic acid, oxaloacetic acid, and alpha-ketoglycolic acid. And we are going to also have ammonia, nitrates absorbed by the plant lettuce roots are going to be converted to ammonia, and ammonia combines with those organic acids mentioned above to form ammonia with the carbon skeletons, and ammonia with carbon skeletons will be converted to amino acids. Then amino acid polymerizes to proteins. Now let us go on how lipids are formed. Now, triose phosphate, they are formed from triose phosphate. Triose phosphate is converted also into extra sugars, or polymerizes. Triose phosphate molecules polymerize to extra sugars, and those sugars undergo glycolysis, forming pyruvic acid. Pyruvic acid is transported into the mitochondria, and it is converted into acetyl coenzyme A. But remember, acetyl coenzyme A, it is the main building blocks of fatty acids. And now inside the chloroplast or the cytoplasm, acetyl coenzyme A molecules, they undergo series of enzyme controlled reactions that add those two carbon units repeatedly to form long chain fatty acids. Now we have formed fatty acids. Now fatty acids now combine with the glycerol to form lipids. Don't forget to subscribe. You can repeat this video too, so that you can get the information more accurately. Thank you guys for following. I'm testing you. For item 2 we are coming, and I'm going to come with I'm going to be providing these items to your last paper, and just know some of my items are going to be in your UNEB paper. I've talked that when I'm ending the video, because I know most people are not going to complete this video, except the few lucky ones. Share this video to your friend.